Hi, well, good morning, everyone. I'm Dennis Akowski, Science Director here at Hudson River Foundation. Thanks for coming. I think we may have a few more people pop in as we go along. I understand we have about 10 or so people on the webinar. And just to go through a little bit of the basics here, the folks that are on the webinar can see us and hear us, but they can't talk directly to us. But at the end of the seminar, we'll post up questions that they can write in, and we'll have a little Q&A session after Steve's presentation. Well, today I'm happy to introduce Stephen Baines. Stephen is an associate professor in the Department of Ecology and Evolution at Stony Brook University. Because you have a joint appointment over in the old Marine Sciences Center, the College of Marine Science and Atmospheric Science. Marine and Atmospheric. You got a couple of old MSRC people in the room right here. Steve has been, I guess, at Stony Brook about 11 years now. He's an ecologist. He likes to look at little things. He's interested in plankton and biogeochemistry of the food web. And what interested us with Stephen was a proposal that he submitted a few years back that was getting at one of our biggest issues in the Hudson River, and that's the zebra mussel invasion. And what he brought to us was a project that he was taking a look at and said, you know, the Hudson River is being filtered by zebra mussels very effectively, and they're sucking down all the zooplankton and phytoplankton and potentially phytoplankton in the river, but maybe they need something else to eat as well. So we thought that was a very interesting project, and he's been working on that the last couple of years, and we're glad to have him here. Steve.
Thank you. And thanks for inviting me. That's why I've been waiting for a long time to talk here. I mean, I think I've been invited before, and I found, for some reason, I didn't come, but I'm here now, and I'm really happy about it. So today, I'm going to try to summarize the big picture elements of the story that Dennis just talked about. I should characterize myself a little bit. I am somebody who started studying plankton, phytoplankton, but I'm also very interested in the things that eat phytoplankton because I'm interested in food webs as well as the base of the food chain. So I've done a bunch of stuff having to do with toxic contaminant transfer through phytoplankton up food chains, and also on these interactions between phytoplankton and things that eat them. And so that's how I ended up dealing with zebra mussels, right? Because they pose a problem in many respects that you don't normally see in the wild. I'll talk about that in a bit. And that's the problem is that they don't starve. In the Hudson River in particular, they don't starve. In fact, there are a few situations where they apparently do starve. And that's a very strange trait for a consumer, something that eats other things. So the subtitle gives you kind of an idea about why I think that's true in the Hudson River at least. And I'll try to explain the elements of that. I do want, so all of this here, every bit of it is from the Hudson River. And there are a lot of people who helped over time, who inspired this work, who helped this work, who helped me in the lab, and who just kept things sane and in control. So, and that's actually a short list. It includes graduate students, undergraduates, high school students, faculty, you name it. So, but a lot of people working on this in the lab. Okay, I'm going to hope this works. Will it? Did I turn it off? It did. Oh, okay. So, basic facts about zebra mussels. People don't know. A lot of people do know, but it's always good to sort of get the fundamentals down. They are filter-feeding freshwater bivalve mollusks. Okay, they look a lot like tiny blue mussels, except they're striped, right? They have kind of the same body form. They're filter feeding, which means that they take water into their gills, strip particles out, and funnel those particles into their mouths and digest things in them. So we typically think of them as being important predators of things like phytoplankton and other things in water. I have to be careful about that cursor thing. Okay.
I don't think it's the cursor. That's the little mouse.
The mouse? Okay, something like that. I think that's what it is. Okay, so they are not native to the U.S. They come from Europe and Eurasia, actually native to a region near the Black and Caspian Seas. And there's actually a whole group of species, a species complex that come from that area. Okay. They evolved actually pretty recently for bivalves. Most freshwater bivalves are very ancient, evolutionarily speaking, in the order of 100 to 200 million years. Okay, and they generally evolved bizarre traits to deal with being in a freshwater environment, running environment. These guys actually look like blue mussels and other kinds of mussels used in marine environments because they're actually really recently evolved from, by comparison, only about 9 million years ago. And they evolved in this region because the sea level rise kept moving up and down, and we had salt water, and freshwater switches, and some things evolved to deal with freshwater in that situation. That's important because they retain a lot of marine characteristics. A lot of freshwater bivalves have lost traits that marine bivalves have, and one of them is their ability to stick to rocks. The other is they have things like planktonic larvae that spread very quickly, and we'll talk about some of the other ones later on. They invaded North America in the 1980s, got to the Hudson River in 1991, and they spread very rapidly through any kind of connected water way since. It's very good at spreading, and they're also very good when they get to a place at growing. They are basically weeds. It's the only way to describe them. A lot of bivalves are hard. They're hard to break. They have shells because they don't want to get eaten. These guys have shells, but the shells are actually quite thin and they're easy to break. And as a consequence, they get quite sharp. They can make these large. They can accumulate so quickly they can make these entire beaches of egg shells and say the Great Lakes, in this case, Lake Erie. And they can have huge impacts just by building up biomass very quickly. They grow very quickly. They invaded the Hudson River in 92, 91. A couple years later, they increased the amount of sort of filtration of the water column by consumers. That includes all the crustaceans in the water column and everything by almost 20-fold just by being there. Okay. And that resulted in a big drop in algal biomass in primary photosynthetic algae in the water column, about 80%. That had downstream effects on things that ate those phytoplankton, including zooplankton, which declined a little bit, and also other native bivalves. It also had effects on the benthos, because actually removing all this stuff that used to be in the water column to the benthos, so suddenly the benthic community changed. And it actually has effects even on growth rates of the fish. So there's a lot of consequences of this invasion. Okay. I used to have a slide which also showed the fact that they just cover stuff. They have a tendency to block water intakes. They'll cover lobsters, anything that moves too slowly. So they're also just very hard on anything that's hard, basically. They'll grow on it. And this effect has not just come and gone, it's been persistent, right? So they invaded in 91, over 92, and basically since then, this change in the ecosystem has persisted. It hasn't changed, okay? And that represents a bit of a problem if you think about it ecologically, because reality, I mean, often consumers like this and baby consumers are this effective at eating their resources, they end up starving themselves out. This is one of the ways you get these predator-prey cycles. Predators show up, they eat their prey, they get stuck, they start to starve or decline, prey come back, and we get these funny oscillations. But that didn't happen in the Hudson River. Okay. And there's lots of reasons to believe they should be starving. Okay. There's a study by Ben Schneider in the Illinois River, which is also a very turbid river. It's a smaller river, much smaller. But he was able to show that if you plotted scope for growth, which is basically just the energy left over for these guys to grow and reproduce after you account for all of their other costs, right? It's kind of the net profit of the household. It increases with the amount of ratio of organic matter to inorganic matter in that river. And actually it turns out to be a very good relationship in that river. And it turns out those zebra mussels behave a lot like the ones in the Hudson River. And so he found that there was this threshold for net growth of about a ratio of about 0.5. The reason why it gets higher is because you get more algae. It's lower because you get lower algae. Basically, this is a measure of the amount of algae in the water. If you look at the Hudson River, prior to the invasion, a lot of the time its ratio is actually above that magic kind of cutoff line. But as soon as they invaded, basically the ratio of that organic to inorganic matter fell off the map. And it doesn't look like the stuff floating in the Hudson River should be good enough quality to allow these guys to survive. There's other things that make you a little bit suspicious of something weird's going on. If you actually measure the amount of CO2 produced by the zebra mussels in the river and add it up, they're respiring, so they're producing CO2, it actually is much greater than the amount of photosynthesis, CO2 fixed by photosynthesis, by plants and algae in the river. So that's an imbalance, right? That carbon has to be coming from somewhere. There's not enough coming in from the river itself. So that suggests there has to be some subsidy from outside of the river that these guys are using to support that kind of respiration, right? Because they can't maintain that respiration if there's no stuff being produced in the river to support it. Well, I've already argued particulate matter, all of it, is not very good, right? And the Hudson River, that includes the bacteria, that includes the plant material, the algae. There's just not much of the stuff that's edible in that mess of stuff floating in the Hudson River. So that other big gorilla in the room is this dissolved organic stuff that floats in the river. It's something we don't typically think of, right, when we think of ecosystems, but in fact, it's a really big part of these ecosystems, right? It composes 75 to 90% of the organic matter, the organic carbon in these ecosystems, right? We don't know much about it. I mean, we're getting to know a lot more about it than we used to, but it's still it's a mess of stuff. It's hugely complicated. It's very hard to characterize. As much as, as little as like 5, maybe 7 years ago, I'd say at least 75% of it was not even understood or characterized at all. So there's this question about what it is. And it's largely terrestrial in origin. You know this from tracers that indicate something about where it comes from. So it's potentially a subsidy, right? If something could use it. The thing that makes it kind of bad candidate for a subsidy, first of all, for a filter feeder, is that it's dissolved, which is a problem if your whole physiology is designed around catching particles and not sort of drinking stuff. That's a problem. Okay. It's also not considered bioavailable. The reason there's so much of it, the hypothesis is that it accumulates because things can't use it, right? So that's why there's a fair amount of it. However, it It is derived from outside the system, and you actually, because there's so much of it, you don't actually have to use that much for it to make a difference to organisms. So there might be a sub-fraction of it that can be used that's actually driving a lot of this. And it's something that isn't constant across ecosystems. It varies a lot. Or among lakes, it can vary 10, 20-fold, right? So this is not something that's going to have a same effect everywhere. Depending upon where a lake is, in the watershed, where it is in the drainage, it's going to have very different values of this. And that means the effects are likely to vary a lot. And not only are they varying in space, over time, this dissolved substance, these dissolved substances have been increasing in many ecosystems over the last few decades, and especially in north temperate ecosystems. This is actually a spot in the Hudson River where dissolved organic matter has increased by 50%, which is, to me, as a graduate student, seems ridiculous. Everyone said it was basically constant, it would never change. That we know is not true. We're not exactly sure why this is happening. There's quite a few hypotheses about it. But the upshot is this terrestrial DOM. There's a kind of a traditional idea that this dissolved stuff that comes in from the water from the watershed, it's basically tea that is made from the leaves on the forest floor, drips down into the lake, actually acts as kind of an augmentation of the food web, right? You basically normally would have phytoplankton growing, they feed crustaceans, they feed fish. Now you've got this DOM coming in, they feed some bacteria, they feed some protozoans, they feed some crustaceans and feed fish, and as a result, you should get more fish, right? And in fact, lakes in Wisconsin, 50% of the carbon can be shown to come from like maple leaves, for example, not from inside the lake. That's a traditional way to think about it, but there's another way to think about it. If only one of these consumers in an ecosystem can use it, it can pose a real advantage for that organism. So in the normal case, I might have plants getting eaten by two consumers, they get eaten by two predators. If I have a subsidy, I can unbalance the relationship between those consumers, right? Suddenly, this guy is only eating these plants. This guy's also got the subsidy and suddenly explode in abundance, maintain large biomass, and suddenly you get a completely restructured food web, potentially as a result. So these subsidies not only are economic, they can actually restructure ecosystems. So why might we argue that this could happen? And the reason we might argue that it could happen is because the relatives of zero mussels can do this. Right? So if I look in the marine environment out here, it's actually marine enough, I can find mussels, clams, actually a number of organisms that can take up things like amino acids directly from water. They essentially suck it up across their gills, across their integuments. Okay? And so that's been measured repeatedly through time. In fact, they're extremely good at it in some cases. Middle effectiveness can remove 70–90% of the amino acids passing through its gills. It's an extraordinarily efficient process for something that's a filter feeder. Right? Okay? So one of the things that kind of counts against that, in general, we haven't thought this is a really metabolically important in most ecosystems, right? Concentrations of these substances, like amino acids specifically, are going to be too low. The rest of the DOM we don't really understand, right? We don't know what kind of role it's playing. And the other argument is the physiological mechanism involves sodium, which is abundant in seawater, but not abundant in fresh water. It makes it hard to work how this mechanism would work in a freshwater organism. I have ideas, I wanted to talk about that much, I have ideas about how that would potentially happen. But like I said, there's more DOM in freshwater than in marine systems. And there are also all these invasive bioducts that recently evolved for marine people. So I'm going to kind of just go through evidence for why I think this is important. Some of it's a little complicated. I'll walk through this, in as simple way as I can because, I tend to think in complicated ways. I have to learn how to talk in ways that aren't quite so complicated. I'm going to first talk about zebra mussels taking up, do they actually take up specific organic substances like we see in marine organisms? Is this uptake actually important, right? So can we assess if it actually makes a difference to these organisms? And finally, what are the ecological implications of this? So first, this. To do this, the first approach we took was to use radiolabeled substances. We used radiolabeled substances because we can use very specific substances, and we can measure changes in concentrations at very small concentrations. So we don't have to add so much that it's different in nature. Right? I can actually measure concentrations, changes in concentrations that are actually pretty close to realistic. I can measure what shows up in the organism. I can measure how carbon in the amino acids gets divvied up in various things. So there's a bunch of reasons to do that. So I did this with a bunch of different substances and just looked to see if they could even take them up. And what you find is, yes, they do it extremely well. It's very similar to what we see in, say, a middle aciduous within a fresh marine environment. This is basically the alanine in solution of the zebra mussel in a little beaker with some water. It's got radiolabeled alanine added. If I trace this alanine over time, within about 4 hours, it's gone. Within 8 hours, I can barely measure it. Okay, and it falls off in this beautiful exponential curve. That carbon shows up in the muscle, in the actual tissues of the muscle itself, and in CO2 in the water. So they're not only taking it up, they're respiring it. So they're using it. Bunch of other substances show the same pattern. You get slightly different patterns with different substances, but you get kind of the same pattern with all sorts of amino acids and fatty acids too. It's a little uncertain exactly what the fatty acid data means, since in these experiments we're adding fatty acids in a way that probably don't exist in nature very much so. But the free amino acids, that is probably what they do with those things in nature. And they're respiring these things to different degrees. Some of these substances they take up and they keep. They make biomass. Some things they actually seem to respire to harvest the energy. So there's all sorts of, they seem to handle these substances in quite different ways. So it's very clear they can do it. They can take all the amino acids up, fatty acids, some sugars to a little degree. Okay. Now, there are possible critiques of this mechanism, right? Radioisotopes allow really quick measurements. They're great. From that point of view, they allow you to measure things very precisely, but they may not measure what happens next. In other words, the organisms could be losing alanine at the same time they're taking it up. But we don't label the organisms, so we don't measure that. So all we measure is what they take up, sort of in gross. It's kind of the gross profits. We don't see the net profits. So what happens when we actually go and we look at dissolved substances in their sort of natural concentrations in nature, and we do the same kind of experiment? We get a lot more noisy data because it's harder to measure these things at these concentrations. But what we see is these amino acids, they decline. You know, when you allow zebra mussels to go through the water. And in many cases, actually, if you don't have zebra mussels, these substances start increasing. It's probably because of bacterial enzymes that are in the water breaking down more complex polyamino acids. So they're basically working against the bacteria that are releasing stuff from these more complicated substances and keeping those concentrations down. So it appears that it is basically net process is happening. And the rates we get for this are actually quite similar to the rates we get for the vegetables, which is the same story people see in the marine world. Okay, so there's net uptake. There are some funny data things here. There's some evidence that there are some floors that you can't cross for some of these amino acids. For other ones, they don't seem to be, they actually seem to drive them down to zero. We don't really know what that is about. Maybe they're releasing some serine, and that's kind of equilibrium. We don't know, but something to look at in the future. Okay, right. So it's kind of just summary stuff. Okay, so what is this important? So we know that it doesn't matter. This is actually always the point where these studies usually fail, because it's very hard to show that it matters, except for very tiny things. Okay, so we tried to assess this in two ways, by using absorption efficiencies. We'll talk about that in a little bit. We calculate based on absorption efficiencies and what we know these mussels do in the river, and concentrations of these substances in the river, and figure out how much they're taking up, and relate that to their needs. Is it large? Is there enough they're taking up? How large is their needs? This is the classic approach people have taken in the past. In addition, I've taken kind of non-classic approach, which is to say maybe we don't know everything they're taking up. Maybe we shouldn't presume it. So what happens if we just give them dissolved organic matter and see what happens just in bulk? And does our answer match up? So we actually had a quick and dirty measurement of amino acids in the Hudson River. It was done by Stony, 3 stations on the river, 1 day. Gave us kind of a number that was in the ballpark, right? So we could use it. We knew what the filtration rate of the mussels are. They're actually very, very typical in the Hudson River for some reason. We knew an absorption efficiency. We can calculate the absorption efficiency for amino acid. We know the concentration of that amino acid. Combining those things gives us an absorption, right? And we can calculate that absorption efficiency. It's basically just the rate at which that substance disappears versus the rate at which they filter food. This is basically a measurement of the amount of water they're pushing through their gills when we give them food that's big enough for them to filter 100% efficiently. And then we can compare that, essentially, pumping rate, if you will, to a removal rate. And what you get when you look at these different substances are different numbers, right? They're not doing the same thing for everything. This is actually a telling thing. I often get this comment, people say, oh, it must be bacteria on the gills. There's no way you can convince me that a bacteria does not want to have take out sugar from solutions, right? I don't know any bacterial community that has that uptake profile where sugars are and acidic amino acids are not taken up, but all the other ones are. What this does look like is what middle cellulose looks like in nature. It's got very specific uptake pathways for different kinds of amino acids that have different kinds of affinities for those things. For some reason, they don't have the uptake pathways in this amino acid. They don't take sugars up very well. They take up polar, neutral, non-polar amino acids 10 to 20%. They take up fatty acids almost 90%. So when we do that, we have those numbers. We can then look at our estimates of these concentrations in the river of amino acids, and we throw some numbers at some of the other data, some of the other substances. We basically calculate that about 20 to 40% of the respiration of these guys, if they're kind of just breaking even. This is the respiration they need to maintain existence without losing weight, right? Just break even. Basically what you like 2,000 calories a day. Well, for me, that's not break even. It's probably too much, but 1,500 calories a day, break even for me. This is our break even carb carbon demand, and it turns out they can get about 20 to 40% of their requirements just by taking up amino acids. Now, that's not everything. It's not nothing, right? But it's not like 100%, for instance, right? So, is that interesting? So, and when I got to this point, I was very concerned because this is basically, I see myself going through the process that we've seen like 1,000 times over 100 years on this debate, right? Back in 1900s, people started arguing, is this important? I've got some bivalves, I dump a ton of sugar into water, they'll take out the sugar. Of course, there isn't a lot of sugar in water. People point out pretty quickly, well, BOM is not sugar, and there isn't that much in it. So I don't know why you can generalize. And other people started getting radioisotopes and showing they can take out specific compounds. But then other people said, well, it's the microbes. They're not doing it for food. They're doing it for osmotic regulation or something like that. And then there was another debate. Other people started using HPLC, these anti-analytical methods, saying, oh, but they're doing it a lot. So it must be important. And other people use the same method to show there isn't that much stuff out there that they're taking out. So you get this kind of repeated kind of sequence. And so it's almost that the whole community has been kind of separated into two groups. People think it's important. There's other people over here who don't, and they don't talk to each other. They'll just come up and do this kind of don't mention it. It's kind of like politics, I guess, right? I'm here doing the same thing. And part of the problem is we keep focusing on individual substances in this debate, right? Specific model substances that we know don't represent the BOM in general. So what is the effect of the bulk BOM on these zebra mussels? So the idea is to expose these guys to water with and without BOM, and then measure some sort of ecological relevant parameter. Does it make a difference to them? So we had an experimental setup to do this. These are painful experiments. I put up those acknowledgments basically as an apology to the people who had to do these things because they take months to do, and it's just this boring, repetitive stuff where you're basically replacing water, measuring mussels, and going through this whole process. And it's hard, and it fails often, and so you have to start again, and they last 2 months, right? Because it takes that long to get a change in the variable we were measuring, which was the weight of the zebra mussels. And they're actually the fastest ones, they're the most bivalves. So we have a system where we would feed them now and then so they keep filtering. But then one set of the zebra mussels, we feed them Hudson River water that had been constantly filtered to get rid of all the particles. And the other, we just fed them water that looked like Hudson River, except it didn't have all the dissolved organic. And what you find is that the guys in the water without dissolved stuff, they lose weight 5 times faster than the guys in the water with dissolved organic matter. It's not a small difference, it's actually a huge difference. Okay? It's not a difference in how they deal with food or anything like that. We measured that, there's no difference there. You can see this is the third experiment I ran. So we did this multiple times to see this result. It takes a lot of reputation to see anything whatsoever. We can actually calculate some of these contributions of BOM to metabolism because we know that soft tissue, the amount of carbon for soft tissue doesn't vary in all of our experiments. So soft tissue loss rate is basically the carbon loss rate, which basically tells you the respiration rate of these guys when they're not eating food. So it gives you a sense of their metabolism, right? So we can do that and estimate the carbon demand for these mussels. And what we find is that they lose weight at about that rate. You calculate some correction because we do feed them a little bit. So their carbon demand is actually, this number is kind of just a number. It turns out to be pretty similar to what people have measured in other studies, so we're pretty happy about that. If you look at the difference between the loss rates between in the two treatments, it gives you an idea about the contribution of carbon from this dissolved substances. And that turns out to be 60% of the maintenance ration for these guys. So it's a bigger number, right? And I think it could be even bigger because we can't keep replacing the water fast enough. These guys, if they can suck up stuff that quick, it's very hard for us to replace the water fast enough to replicate what happens in the field. So this DOM concentration is much larger than other past estimates for adults anyway. It's 2 times that more than we estimated using these concentrations of substances in the field. So that's a puzzle, right? Did I do something? Nope. Am I playing the wrong thing? Okay. Why does this matter? Does it matter? Okay. So if I plot, zebra mussels takes a long time for them to starve, to die of starvation. Actually, they have to lose 75% of the weight before they die on average. So we can calculate how long it would take them to die, to lose 75% of their weight, given these numbers. And it turns out that if I, with the DOM, they essentially double their lifespan if there's no food at all. If I give them just a starvation ration, which is basically what they have in the Hudson River, these guys in the DOM can live 7 years without, they're basically making them more. It's something else in 2 or 3 years. So they can't starve in the Hudson River. It's impossible for them to. Right? They're designed not to do that. Okay, I think I got this right. But we have this problem then because we don't know why the effect is that big. We measure the amino acid effect, but this effect seems to be bigger, right? Why is this? What is going on? And that is kind of an ongoing problem. It's what we're kind of working on now. There's potential for uptake of other compounds that we haven't accounted for. There's also potential that concentrations of amino acids that we've underestimated them badly, and there's a good reason why we might be, because we're taking these samples from the middle of the river at the surface, and that may not be at all what these super mussels are actually experiencing right down near the bottom. But it's a hard sample near the bottom. Other possible compounds, I'll just go through some of these. We've looked at polysaccharides and polybeans. The answer is no. Basically, this is the initial, that's a control, that's what the zebra mussels are there. Sometimes actually a little bit more when you have zebra mussels in there, and they actually sort of excrete stuff. But basically, even if I look at saccharides or polysaccharides, the total saccharides, I'm not getting the bulk effect. And that makes sense given what we've seen in the radioactive stuff. If I look at combined amino acids, these are polypeptides, right? Again, initials, 4 hours, 8 hours, there's no pattern, I can tell you this. In fact, and these are the controls over here, there's really no pattern at all. There's no evidence that they take up polyamines in any kind of net sense, not from these kinds of measurements. These are noisy measurements, mind you, because these are the actual bulk measurements that we did this on, but there's no evidence that it's important. Other possibilities, the fatty acids are an interesting thing that could be out there, that they're energy rich, but they're difficult to deal with. They're complex in many cases. They're complex other substances. They may not be really freely available like in our experiments. There's colloidal material, stuff that kind of forms little tiny particles that they may be able to collect through its electrostatic forces on the field and essentially eat it. But we haven't actually looked at that yet, and it's actually really tricky to argue that that's happening in freshwater situation. And then there's the possibility of other substances that we don't know because DOM is such a strange substance, a collection of substances. What about these free amino acids? We have a lot of data on these now in the Hudson River. We've got a lot more data. And one of the main things that we see that's frustrating and also probably very interesting is that all three amino acids, this is total amino acids, single amino acids, free in the water, vary 10 to 20-fold in some cases over the course of the year. And that is big. That's big for these zebra mussels. If that number, we estimated the number is about 0.2. If that number ever happens on a regular basis, it totally changes all of our calculations. And suddenly it becomes an issue. It's definitely something that seems to happen more upriver than downriver. Yeah. So the other thing, this is going to make it hard to assume that the dissolved amino acids are just a constant fraction of VOC. So we can't just measure VOC and assume a constant fraction is getting taken up, which is frustrating. What am I doing with this? I think I'm maybe running out. Okay, composition varies a lot. There are a lot of different amino acids. I don't want to, I showed this picture because it's pretty, it doesn't have any information in it, from what I can tell. Okay, I've actually looked at a fair amount. There are no systematic differences between sites and seasons that are like regular. There are a few amino acids that really dominate. We'll talk about those. There's 7 others that kind of only dominate rarely. They're just suddenly there, and then they're not in any other sample. Okay, and these are basically serine, aspartate, glycine, alanine. All things, well, these 3 in particular are taken up very efficiently by zebra mussels. So most of the amino acids, the vast majority of it is very available to zebra mussels. It's out there. There's also a varying amount of these things. One of the things that is kind of useful about this is that a lot of these amino acids are really tightly correlated to each other. That means they're proportional to each other, which means the kind of bioavailability doesn't vary much from site to site, really. If I know the amount of amino acids, I can basically tell you what zebra mussels will do with them. I don't need to know much about the composition because it's pretty consistent. Okay, so, right. The last thing I wanted to talk about was this potential impact. So we talked about the effects on the zebra mussels. What are the potential impacts on the ecosystem? That's the thing that really interests us. And this calculation is a little backward. It seems backward to a lot of people, but it's based on a very simple assumption. The zebra mussels, like a lot of consumers, should suppress their resources, their food resources, to the point where they have kind of zero net growth. That's kind of an assumption of a lot of population models. So they will grow until they really can't grow on their resources anymore. Since they're subsidized, they don't starve, so we don't get these big crashes in cycles. So we'll have that, so I'll say, okay, they're going to depress food to that level, calculate what this value is with and without this subsidy, and then see how this compares to the average quality that we're seeing for pre and post. I won't go through the numbers, but there's a bunch of numbers associated with this. It's all from the literature, and some of the stuff is from our study. But basically, the upshot, it's a terrible graph. I don't know why I got worried like that. Is that if we calculate this for the Hudson River, we see these zebra mussels. That break-even point is actually lower than the current concentrations in the Hudson River, right? Which we'd expect if we were right, because they don't only have to just not, starve. They have to actually also compensate for predation and death. So actually, they're going to need a little bit more of the population to maintain in the ecosystem. So this actually means the estimates for energy inputs are pretty consistent with this data that we have on the effects of the zebra mussels in the Hudson River. If we don't have BLM subsidies, basically most of the time they're likely to starve because most of the time the annual average chlorophyll dose isn't enough to keep them alive. And in fact, we calculated the population level numbers would be way up there. So the upshot is that the calculated benefit of the DOM, the mussel itself, is actually pretty consistent with their ecosystem effects, and the effects we've observed on biomass of plants in the Hudson River, of algae in the Hudson River. They would still have an impact without the DOM subsidy, but it would not be nearly as large. I mean, it's depressed concentrations, maybe 40, 20%. It would be nothing like we see now. It will probably be a lot more variable in time. And that's basically all kind of, if you think about this, it kind of inverts our understanding of the way these subsidies work in ecosystems. I mentioned before that we have this terrestrial subsidy coming in, and typically we think of this actually augmenting food webs, increasing production, you know, at the top level, the point of the, say, fish that we might want to eat. But in the case where you have zebra mussels, that's a very risky thing to do an animation with this pointer. You get a zebra mussel invasion, right? Suddenly you change the movement of this stuff from going this way to going this way, right? Now, normally the zebra mussels would starve, and eventually their populations would moderate, and you might not get a very big effect or variable. But with this subsidy, what we're getting basically is the ability of these zebra mussels to maintain biomass and persist and keep this total reorganization of this ecosystem in place. Okay. That's what I argue. Okay. Small fish, as a result. I knew there was going to be something else. Right. So that's it in a nutshell. Kind of a review of the position. It's not necessarily something unique to zebra mussels, we don't think. In fact, for those ecosystems where people see 50% of the carbon from trout being trees, people don't actually understand how that carbon gets there at this point in time. It doesn't kind of make sense that it gets there through the standard mechanisms. People think it actually might get in through the consumers themselves taking it up. And it's hard to exactly know how that happens right now. It's one of the big questions. So the implications of that are potentially big too, if it changes the way we see the effect of consumers on these ecosystems. And that's it. I didn't want to dwell too much. Yes.
If the DOM is coming primarily from leaves washing in, why is it so rich in amino acids? I don't think of leaves as being rich sources of protein and amino acids.
No, they're not. Well, what happens is the leaf material comes in, but then it gets worked over by bacteria in these systems. So you don't get tannic acid getting into the zebra mussels. What happens is that stuff gets decomposed by funguses and bacteria. They alter that material, turn it into proteins because they have to have proteins in order to survive. And then they release those, and when they get eaten or when they die, and that's what ends up in the water. So that's where the polypeptides come from. They're almost all processed terrestrial stuff, but processed through the bacteria in the river.
It's clear that it's really mostly coming in from terrestrial sources rather than being recycled in the aquatic system.
I will say that we aren't entirely sure about the amino acids, but the bulk of the dissolved organic stuff is definitely from the terrestrial. It has a completely different. You have to sometimes go to particular ecosystems where you have nice contrast between what's in the river and what's in the watershed. But when you do that, you find that that DOC looks like trees almost every time. Yeah, and kind of makes sense.
Well, if you looked at the carbon-14 labeled pseudo-feces, do you see any evidence of
Well,
label in the pseudo-feces?
so we've done that for other purposes. We didn't do this because they weren't supposed to. Oh, carbon. So you're saying? Well, they don't have. We're not giving them C14 in particle form. Right.
Okay. Right. Yeah.
Right. So it is possible. If we were feeding them at the same time we were trying to label with the amino acids, that you would get some of the radio label into pseudo-feces and things like that.
The other thing is that you're talking as if there was these mussels maintain a stable biomass over the year, but most mussels have major reproductive cycles that affect their biomass.
Yeah. So what I mean to say when I say they maintain biomass is they're maintaining well above what we expect them to if they were just depending upon food in the river, the particles in the river for food, right? So they do vary in terms of biomass because there's a seasonal reproductive cycle. Usually starts probably happening now, as a matter of fact, and sometimes the second burst is late summer, early fall.
It's pretty much the way it is with other mussels as well.
Yeah. But they also have kind of population dynamics and some demographics that change over multi-year time scale. There's usually these big guys that come up and kind of take over, and then they kind of starve out the small, or something happens. Some people argue they eat the larvae, so they actually cannibalize, but then they reproduce, and then you get a bunch of small guys forming a cohort, and then you get this kind of repeated cycle of small versus large in the population.
You're suggesting they can live for 7 years, but they actually only live for 3 years, is it?
Oh, I don't think these 3 would be generous. I think they probably live a year and a half, at most on average, maybe a year. And that's only after they get to a certain size, you know. And lately there has been, so the first couple of years of the latest version of this project, it kind of disappeared from the river. And it appeared like something was killing them. In all the places we used to collect them, it was a real challenge. I actually had to go out and find them myself, because my coverage could not figure out where to go look for them. Yeah, so they don't live very long. It's not exactly clear what kills them, but you can estimate that their death rate is pretty high. It's not surprising. They really are cheaply made. But there are not a lot of native things that are designed to eat them. In Europe, so they do have crash boom cycles in a lot of places. But those places are almost always places that are shallow. So when they invade, they become really abundant. And what happens is suddenly migrating birds find them. And once they find them, they all show up at the lake and they're just creaming them. And then you'll get these crashes. But in the Hudson River, that doesn't happen because most of the river is not available to those guys. So people talk about blue crabs, maybe. Yep.
Is it possible that part of the DOM is something that's like not traditionally considered nutritious, like some sort of zebra mussel stimulant or something they're using?
Stimulant? Oh, you mean like a steroid or something? Well, so people talk about. Well, so the fatty acids would be the natural place to look for that, right? Because there's a bunch of essential fatty acids which people relate very strongly, especially to success-free development for the larval stages, which are untonic, but it turns out they really need essential fatty acids in their food in order to metamorphose. And so the population maintenance is very likely that there's some environment like I don't think you can get around the basic requirements of energy, right? And if you respire a certain amount, you have to balance that. There's no way around it. So you're not going to get around that part of it. There are definitely situations where specific substances have consequences for life stages and more.
Given the temperature differences over the seasons of Hudson River, there will be a significant metabolic differences in the mussels. Is it not affected by the cold temperatures?
Oh yeah, they're affected a little bit. I have to say, when I go out to see them, they haven't been wiped out by ice, right, which is a common problem here at the surface. When I go out to see them in the early spring, they seem to be quite happy. I mean, I don't exactly know what they're doing. They tend to be cold. In Europe, they tend to be more colder water. People tend to think of them as being more, when they raise them, they'll raise them at 16 degrees Celsius or 12 degrees Celsius. They think that's more natural. So they might actually have a high temperature limit, but they haven't shown. I mean, there is some evidence that at above a certain temperature, their respiration starts skyrocketing and overshoots muscle levels. They hold up, but you haven't really seen that in terms of their distribution. It seems to constantly progress into these more southerly environments, and so it's not clear. It may be evolving. I don't know to deal with that.
There's a question from the webinar.
Can you read the question aloud?
Can you read it out loud?
I am trying to, so this is the question. Do you then predict that a watershed management effort to reduce the input of organic matter received by the river would positively impact river mussel populations? And then, I guess the idea here is if we changed the management of the watershed in a way that changed the flow of organic matter off the watershed into the river, I guess the idea is reducing it somehow. I guess, and I'm not exactly sure what management action you would take. If you're doing a lot of land use transformation, it's possible you could actually cause a lot of a lot of the soils into the river. It would be kind of a short-term pulse, but it could happen. But I think, and there's certainly a lot of the UM derives from wetlands. You want to actually remove wetlands. I'm not really sure that's always a good idea. So I'm not entirely sure what management actions you would take to reduce DOM flows into the Hudson River, because all of this stuff gets into the river. It's not useful when it gets there, but because it gets worked over by the biotosphere, that's it's very hard to figure out how to change that flow in such a way that it was going to be able to change the impacts on the zebra mussels and the rest of the ecosystem. This is actually kind of, in some ways, kind of a depressing story, because it doesn't suggest obvious ways to manage. What it suggests is ways of thinking about how population variation is likely to respond to different environmental changes in ways that we maybe wouldn't have predicted a priori. And so maybe gives us some tools to adjust to those unusual responses. If you could find what the mechanism is that they're using to take stuff up. So I didn't talk much about the physiology of this because, frankly, it's physiology of UM uptake and amino acid uptake is actually pretty controversial in some areas. But in marine systems, the idea is that the mussels have lower sodium concentration in their tissues than the environment, and actually work to maintain that gradient. And so what happens is sodium is always going down gradient into the organism through channels. And the argument is that what happens, the reason they're allowed, they can take up acids at such a low concentration, they're essentially coupling that downward transport of sodium ion to amino acid. So there are actually evidence that there are co-transporters of those things. In fresh water, there's no sodium in these systems. And most freshwater mussels and plants have evolved to be extremely tight with respect to maintaining ion. They don't want to lose them. They're important for biological activity. Zebra mussels are weird. They have the lowest internal concentrations of ions of any mussel that I think in freshwater has been measured. And they're leaky too, so they leak these things all the time. So they're constantly deliberately kind of taking them up to compensate for that. And one of the things I think might be going on is they're essentially using the same mechanism, just as an accident of having to suck up the sodium. They just, it happens to be attached, the same mechanism in marine systems, sort of just break that amino acid in. If you could find a way to break that uptake channel, I don't know if it would kill everything else, but that would be one way you think this would tell you how to manage zebra mussels better.
Well, the early part of your talk, you had some slides showing the ratio of inorganic to organic material, and I guess I got from there that the higher amount of inorganic material, the harder it is for these guys to survive. Is that kind of the takeaway?
Right. So the zebra mussels in the Hudson and in the Illinois River, a lot of mussels will adjust their filtration rate to food quality isn't very good. The zebra mussels don't seem to be doing that. They're just cranking as fast as they can. They're acting as if they're worms in mud. They're basically, the strategy of a worm in mud is to eat as much as you can, as fast as you can, get as much as you can, the good stuff out of all the bad stuff there. So you seem to be having that strategy, right?
Is that good or bad? I mean, is it better than shutting down?
Well, I think in this case, what it allows them to do is, I think that it may be the only reason that makes sense is because of this, of the DOM uptake. Because if they constantly move water past their gills, they're constantly getting a new source of this stuff. And so essentially, it may improve the supply of the subsidy. And so basically, you're stuck with this choice, right? They're in bad food, I shut down, I can't eat. It's not like it's going to get better soon. If you have access to the subsidy, what reason is there to shut down, really? It really makes more sense to just keep cranking as fast as you can. And they're a little unusual in terms of bivalves for this. I mean, I've seen them you can actually have the Hudson River water just look terrible, and they will just clean it out in hours. And that just cannot be good for them, I keep thinking, because most mussels are shut down in that kind of situation. But most mussels slow their filtering so they can sort particles on their gills more effectively. They pump that much water in that all of this junk is very hard to sort.
Well, I was wondering about it, like when Lee and Irene, those episodes where you had very high concentrations of suspended solid, whether that could be deleterious to the zebra mussels at all.
Yeah, I mean, it'd be interesting to look at. So I would argue, one of the things I want to look at is this variable amino acid data set we have to figure out when they took the samples relative to the tidal cycle and see if we can say, oh, well, every time the amino acids were high, and in fact, all this stuff had been stirred up from the bottom on a daily basis. If there is a daily pulse of this stuff from the bottom, it may actually be a way to kind of really ramp up this mechanism. So, the Hudson River is very dynamic. It's dynamic on a daily basis. It's dynamic on that basis. I don't know if they can actually deal. It'd be interesting to ask if they can deal with like 5 days straight of it. What happened, or whatever, how many ever days straight it was during Irene. But I've not seen any indications they slow down.
about at night?
Hmm?
Is there a light cycle related to feeding?
They do, in most of the Hudson River is dark. The light only gets about maybe a meter in, right? So the vast majority of zebra mussels are pretty dark. They feed about 24 hours, maybe 18 hours. They regularly shut down and then open up again. It's kind of a periodicity for feeding. So when we do these calculations of filtration rate, in the Hudson River, we have to count the fact they're not always filtering the rate of return. They're going full bore. So, I don't know if that answers your question.
Yeah. Early on, you had a slide, one of your very first slides was change in second disk data.
Yeah.
And the benefit, the feeding rates as well, in relation to previous questions.
I think that's changed very little in the Hudson River. And the reason is because most of the, as I was saying, the river's dark, not because of phytoplankton, not because of the algae, but because of the stuff that gets stirred up from the bottom. And because it's a tidal system, that happens a lot. So you don't get these situations where it gets in the water and then stuff settles down. And so second disk did change. It was changed measurably. It was like pretty minimal. As I remember, it was like 20%. So it moved from something like half a meter, a little more than that, to like almost 75% of a meter. So that's not a whole lot, given that the river is actually pretty deep. It's got an unusual profile for a river, yeah.
Have you, not sure if you checked, have you noticed any differences in the fish species population in terms of their decrease in size over in the
I do not know that question. In terms of, so the data I showed you was specifically about reproductive rates, I think, and recruitment. It wasn't necessarily looking at condition. And I don't know if anyone's looked at that, to be honest. You know?
Well, there's a big Dave Strayer and Kathy Howe, I think, right, you had part of it at least on there.
Yeah.
You talked about that.
Right, and it was specifically for certain kinds of fish. So there are fish that are planktonic at some stage. They showed an effect. The ones that are, some of them actually increase in growth rate. So you're basically shifting the whole food web into this methane pathway. The way it's water.
And oysters and zebra mussels, how well do they get along together?
They don't usually cohabit because oysters are usually saltier sections. So zebra mussels in the Hudson River. In Europe, zebra mussels are more salt tolerant. They'll get to like 5 per mil sometimes, which is pretty brackish. 3 per mil. We don't see them anywhere near that in Hudson River, right? They just don't seem to be able to deal with it. I don't know if that's to do anything with the mechanism, but they don't do it. And so the oysters are all in the brackish and salt water portions. The zebra mussels are really unusual. There are more species like this. A lot of these invasives that have come from places that are these species generating areas that have these recent marine backgrounds are often a lot of the freshwater invasives that we see because they have all these characteristics. And the good question is, why did everybody else lose these characteristics? Because almost there is no freshwater from the traditional freshwater families being out that I know of that have a truly planktonic larvae. They often do things where they will nourish basically saying, oh, the only guys to survive that trip are guys that can do something like this, because most of their food dies on the way there, right? So it'd be very interesting to compare the mussels here, the ones in Europe, to see if there's a big difference between them in this trait. I've never, that's a good excuse.
Okay, one more.
So the Hudson's somewhat unique for a freshwater river, a lot of it flows two ways.
Yep.
Which is ideal for this mussel, as opposed to the native mussels, which are really.
Right, so that's one. Right, that's one of the arguments for losing planktonic larvae, because making planktonic larvae means that they get shoved downstream and you don't have a choice in the matter. They're going to go one way, and eventually what's going to happen is you're just going to walk to the ocean and then run into salt water and die. That's the whole hypothesis. You know, they live in all sorts of river systems that aren't like the Hudson, and they haven't shown a tendency to like go and disappear. It may be that a certain set of conditions have to exist to really make that mechanism work really well for a longer period of time. So, yeah, I don't know. Well, thanks, Steve. That was great.
Well, that was our last seminar of the spring season. We'll be putting together, a list for the fall, so stay tuned. And if you have any suggestions on speakers, let us know. Hey, thanks, and thanks everybody on the webinar.